Okay, good afternoon, student. No, not good afternoon, good morning. I hope I'm
Good morning, ma'am.
so. I guess few are still to join, but most have joined. Sam was not in a previous class also.
He was there in last class.
He was there. Okay, fine. Okay, so I think we can start and others can join. So last time we did micropropagation and we studied that how it can be done by different methods such as callus culture or a meristem culture, right? And then how different stages are there, what are the advantages of a micropropagation and disadvantages also of a micropropagation. So advantages, we learned that it can be used to regenerate number of a plant in a short period of a time, and it can be used in afforestation, right? Right, because we know the deforestation was happening at a very enormous scale. We are using a lot of plants, but we are not replacing them, right? So there we can use this micropropagation technique to again grow number of trees, so we can produce a number of those plantlets or a tree plantlets or a starting material by this micropropagation technique, and then it can be used there, right? So today, in continuation to that, we are going to study another thing which is known as a soma clonal variation, right? So soma clonal variations, they are certain genetic variations basically which the tissue culturists they observe in a plants which were derived from a somatic tissue culture in vitro, right? So otherwise what was expected of these somatic, if we are producing any cells from a somatic cells, right? So they should be identical, so we are not expecting any kind of a changes in those cells. That is the soma clonal or somatic tissue culture is basically all about, right? But this was to a surprise that the plants which were grown, generated by a tissue culture cycle, they were showing some kind of a variations, right? So they studied it that why this is happening. And yes, this discovery, somaclonal variations are being helpful in a certain ways, or they are being exploited for a beneficial uses. And then there are certain drawbacks also, right? So the term somaclonal variation was given by Larkin and Scowcroft in 1981, and it was basically for the changes or variations which were generated as a result of use of a tissue culture cycle or a plants which we were growing through a tissue culture. So somaclonal variation is, if we go for a definition, is basically a genetic variation which is observed among progeny of a plants which are obtained after somatic tissue culture in vitro conditions. So plants which we are getting after the somatic tissue culture, they are showing some genetic variations. So usually, as I told you, the plants or progeny plants which are to be regenerated from the somatic cells, they should be identical, right? Theoretically, they should be identical. However, yes, variation might occur in a number of progeny which are known as the soma clones, and they are genetically variable from their explant. Explant is the starting material. So we thought that the plants that we are generating from this explant, they should be similar, but here some kind of a variations were noticed, right? So initiating explant for a tissue culture, excuse me, cycle may be, we know explants could be different types, right? It could be embryo, it could be root, or it could be leaves also, or it could be from the protoplast also, right? So all somatic tissue culture can result in a soma clonal variation. So that means whatever explant we are using, from any kind of explant that we are using, the variations can be observed or they are being observed, right? So somaclonal variation is a phenotypic change which is as a result of a chromosomal rearrangements during the tissue culture. So when they look into it, the experimentally they try to see that what is happening, what kind of changes are happening. So they found that there are certain chromosomal rearrangements were done or they happened during the tissue culture, which led to the somaclonal variations, right? So here they label the plant which are regenerated from a tissue culture as a R or a R0 plant, right? And then the self-fertilized progeny जो होती है उसको R0 plant as a R1 हम represent करते हैं, then R2, R3, R4, a subsequent generation we name them, right? Subsequent generation which are produced by self-fertilization are called R2, R3, R4, etc. So this the people who discovered this Soma-clonal variations, Larkin and Scowcroft, they referred to regenerated plant as a SC1 and the subsequent plant as a SC2, SC3, SC4, and SC5 generation, whatever generation. If it's the third generation, SC3. If it's the fourth generation, SC4. So this is how multiplication is being done, right? In a plant breeding also and a plant tissue culture also, right? So changes in a chromosome structure. So they found that in contrast to changes in a chromosome number, there are more cryptic. Cryptic means hidden chromosome rearrangements. They could be responsible for a genetic variations, right? So they reported that they found that there are certain translocations which have been reported in potato or oats or a ryegrass, and there are certain published work which shows that there are certain chromosome deletions or duplications or inversions or a different type of a, you know, these are the changes in the genetic makeup of a or in the chromosome due to the shifting of a chromosome or a deletion of a chromosome, right? We all have, you must have studied this in genetics also, right? So there are certain changes, maybe could be due to the duplications, could be due to the chromosome deletions, and these changes were found in the regenerated plants such as rye, grass, or a barley. So they found that there are certain type of the changes in the genetic, right? So then they found the gene amplifications and the nuclear changes, right? So there are different studies which shows that nuclear genes are also affected by a tissue culture stress. So stress, right? They are using the term stress here, right? So when a plants, they are grown under tissue culture conditions for a longer time, or they are being multiplied for a longer time via plant tissue culture uh, in in uh, in a proper artificial environment, so they also develop certain kind of a tissue culture stress, right? So it has been seen that heritable, quantitative, and qualitative changes are observed in the nuclear DNA content of a double haploid Nicotiana sylvestris, which is obtained from pollen cultured plant, right? So these plants, they contain generally increased amount of a total DNA and increasing proportion of a highly repeated sequences. So they observed the nuclear changes also, and they also observed the increased amount of a total DNA in these plants which were grown by this tissue culture method, right? So they found that both AT and GC rich fractions, they are being amplified in a tissue culture derived plants. Then there was a person, this Duranty et al. In 1983, he showed a similar amplification of AT and GC rich fractions in DNA from this Nicotiana plant, right? Within the hours of a culture. They found that there is an amplification in these particular AT and GC rich fractions. So these studies suggest that presence of a differential replication process during early stages of a de-differentiation, which takes place during a tissue culture. Now, it has been seen that plant cells, like those of other eukaryotes, they can increase or decrease the quantity of a specific gene product by a differential gene amplifications also. So there are a lot of studies which were being done to see that why these kind of variations are happening, and they come out with this result also, right? For example, they found that a ribosomal RNA gene amplifications are now known to be very common in a wheat, rye, and tobacco, right? And then in flax, ribosomal DNA is known to alter directly in response to environmental and the cultural pressures that are happening in the plant tissue culture. Then there is a reduction in a ribosomal RNA gene, which was found in potato plants, which were regenerated from the protoplasm, right? So additionally, they both structural rearrangements within the ribosomal DNA and methylation of a nucleotide sequence of a ribosomal DNA has been observed, right? Methylation, DNA methylation, right? Which is an epigenetic change we call a DNA methylation. So they observed that also, right? So what was the basis of a somoclonal variation? They just a moment, huh? Okay, so there are some basics of a chromosomal rearrangements which resulted in after a lot of studies and a lot of experiments and a lot of research, they found that there are certain bases for this chromosomal rearrangement which is taking place. So it could be a karyotype changes, it could be changes in a chromosome structures, it could be a single gene mutations, it could be cytoplasmic genetic changes, it could be mitotic crossing over, or it could be gene amplification and nuclear changes. Then the transposable elements such as jumping genes, right? So they can move from one part of a chromosome to the other part, or the DNA methylation. So they came up with these as the some of the basis of a chromosomal rearrangement which are resulting in a somaclonal variation, right? So first is a karyotype changes, right? So plants with the altered chromosome numbers, it was reported by a lot of workers or a lot of researchers. They found that polyploidy was the most frequently observed chromosome abnormality, right? For example, aneuploidy in the oats or a potato and a barley. You understand the term polyploidy. What is polyploidy? Who will tell me? I hope you know these terms, right? In genetics, you have studied. What is polyploidy? Hurry up.
Ma'am, it's a kind of conditions of the
Uh-huh.
cells in which they have two paired sets of chromosomes in some
Yes,
organisms.
yes, yes.
Yes, ma'am.
Okay, fine. So normally we have a two sets of a chromosome in the organism. So if there are more than the two sets of a chromosomes, that is called a polyploidy, right? Or it could be the less than the two set of a chromosome. So these are the abnormal conditions, right? So they observed that the polyploidy was very frequently observed phenomena or abnormality, and aneuploidy in a oats or a potato or a barley, right? So the change in a chromosome number in a variant plant is usually associated with a reduced fertility and with altered genetic ratios in the progeny of a self-fertilized plant, right? So this could lead to these kind of a changes. Then they found that the single gene mutations, you all know the single gene mutations, right? So single gene mutations, again, they have been studied by several workers, and they found that recessive single gene mutations are suspected if the variant does not appear in R0 or SC1 generation. But then when self-fertilized R1 or SC2 progeny is segregated in the 3:1 ratio for the trait of interest. So single gene mutations mean changes in a single gene. There is a mutation in a single gene of a particular plant, and that would lead to changes. So this type of analysis has been reported for the, again, a Nicotiana plant, rice or a wheat, etc. All these plants, they have reported this. Then they said that there are the cytoplasmic genetic changes. So they have done a lot of work for this on the maize, and the lot of studies are there which shows that mitochondria is the determinant of male sterility, and Bacteria made is T toxin sensitivity in the maize. So it is there somewhere in the mitochondria. The original lines they used had all the Texas male sterility cytoplasm. So due to which the plants, they are normally highly susceptible to this particular disease. So how we can change the picture other way around by using certain plant breeding experiments, right? We can change this. We can make this resistant to this particular disease if we just made one change in this particular gene here, right? So when the selection pressure was applied by subculturing callus, what is selection pressure, right? Selection pressure ये होता है, we keep on culturing or we keep on breeding the plant till to the level till we achieve the desired result, right? तब तक हम उन्हें multiply करते रहेंगे या breed करते रहेंगे जब तक हमें desired result नहीं होंगे, उसको बोलते हैं selection pressure, right? So when selection pressure was applied by subculturing callus culture in the presence of the toxins, so some of the plants which were regenerated, they were found not only resistant to the toxins, but they were fertile also, right? So when they did the restriction analysis of a mitochondrial DNA, they found that significant changes had occurred in mitochondrial DNA which were derived from the cell culture. So there were a lot of changes which were observed there. So molecular analysis, which was again based on restriction endonuclease digest pattern of a mitochondria and chloroplast genome, which were derived from protoplast, they also revealed significant change in the mitochondrial genome but not in the chloroplast chromosome. So you understand this now. When we want to see any kind of a changes in a particular DNA or in particular genome of particular organism or any particular gene, we follow a restriction fragment length polymorphism, RFLP. We do RFLP, right? So what we do, we amplify them, and then by using certain common enzymes, we restrict them, we digest them, right? So अगर different pattern दिखाई देगा सारे genes में, for example, if we digested them with the EcoR1, all the genes. So if they are showing a different digesting pattern, that shows that there are certain changes in their genome, right? So this is how we study the changes also, that if there are some changes which are being incorporated in the DNA or in a particular genome. So how do we study? So RFLP is a technique. Restriction endonucleases are being used in this technique, right? Or a random amplified polymorphism. So all those techniques are basically very commonly used in the breeding experiments also. Then it was suggested that the variations which resulted from substantial DNA sequence rearrangements, and which could be the deletion or addition or any kind of a recombinations, and which cannot be explained by simple point mutations. So these changes were not due to the simple point mutations, right? However, point mutations can also occur in the genomes of the organelles of the regenerated plant. So variations in the chloroplast DNA have been studied in tomato and the wheat somaclone. So they have studied in some other plants also the variations in the chloroplast DNA, right? So next was the mitotic crossing over, right? So mitotic crossing over can also result from some of the variations which are detected in the regenerated plant. So again, these can account for recovery of a homozygous recessive single gene mutations in some of the regenerated plants. So somatic cell sister chromatid exchange, if it is asymmetric, it can lead to deletion and duplication of a genetic material, and that can lead to the further variation in a particular plant. So this can happen due to the mitotic crossing over. Then we have transposable elements, right? So there are these are basically jumping genes, basically, for example, right? So transposable elements, they can be responsible for these variations. For example, heterozygous light green soma clones with high frequencies of a colored spot on a leaf surface have been detected in a Nicotiana tabacum, and for Nicotiana tabacum plus Nicotiana silvestris somatic hybrid, right? So this somatic hybrid was having an unstable pattern of inheritance that would be consistent with the unstable gene. So sometime, some genes, they can jump from one place in a chromosome to the other place also, right? So that could lead to some variations in the plants. Then a causal relationship between genetic instability, possibly related to a tissue culture, induced transposition and somaclonal variation was speculated. So they genomic changes in a MAGE line and mutations in the tobacco lines were observed due to activation of transposable elements under in vitro conditions. So they have studied this in under in vitro conditions, that genomic changes, they were observed in the maize line due to the activation of a transposable elements. Then DNA methylation. So DNA methylation, or it is also called epigenetic changes, right? So it plays a very important role in regulation of a gene expression and its implication in somaclonal variation. So many genes show a pattern in which the state of methylation is constant, but sometimes it can vary at other places also, right? So a majority of the sites are methylated in tissues in which the gene is not expressed, but non-methylated in tissue where the gene is active. So active gene may be described as an unmethylated one, right? So DNA methylation, they said, can lead to some kind of a variation. For example, a reduction in the level of methylation is some part of structural changes needed to permit the transcription to proceed, right? So important changes in methylation level of genomic DNA in course of a de-differentiation and somatic embryogenesis have been reported for higher plants. So they have reported in some somatic embryogenesis or in the course of a de-differentiation. So there are certain important changes in the methylation of the DNA. So wherever the gene expression and its regulation is playing a role, DNA methylation becomes important factor for getting the somaclonal variant. So DNA methylation can also lead to this kind of variations. So instability of a gene can arise through the methylation pattern of any particular gene, right? So causal relationship between genetic instability possibly related to tissue culture induced transposition and hence somaclonal variation. So higher plants में क्या होता है? Many controlling factors they act together to achieve desired gene regulation, right? So if one of these level of control is provided by adding a small tag called methylation, but we have methylated a DNA into, for example, we have added a methyl group to the bases, right? For that. So this methyl group tag sees and it is written as a MC, methylated, right? So we designate them as a MC. So simpler organisms such as many types of bacteria and a single cell yeast usually do not use methyl group tag CS in regulating their genes. Some bacteria, but not all, use methyl tag as MA for this purpose, right? So however, most bacteria, they have a specific pattern of MC and MA for this purpose, for signaling their DNA called methylated DNA or DNA methylation process, right? So this is, we don't need to understand this. So this was just to make you understand the DNA methylation. The major fact is that the DNA methylation in a somaclonal variants or plants which we are getting through this somatic tissues can lead to the somaclonal variations in the plant, right? So this, you just need to study this. Let's not go for a thing. So what are the applications of this, right? So this discovery was when first it was found out, it was reported as a negative thing, because when we are generating plant through a plant tissue culture or through somatic tissue, we definitely want them to be similar. We don't want any variation. So somewhere it was regarded as a negative point. Somaclonal variations were never desired. But then within a period of a time, people started to see that why this is happening, and then how they can exploit these kind of variations and how these kind of variations can be beneficial, right? So there comes the application of the somaclonal variations. So they can sometimes lead to production of a novel variants also. Novel means new variants, plants which were not present earlier and which can be useful also. So an implication of a somaclonal variation in breeding. So this technique can be exploited in a breeding to generate a novel plants or a novel variants, right? So they can arise and they can, if the properties or characteristics are useful, they can be used agronomically, right? So a number of breeding lines have been developed by using somaclonal variation. For example, enhanced scented geranium variety, which was named Velvet Rose, has been generated by this method. So an example of heritable somaclonal variation is the development of pure thornless blackberries. So they have done that also. So definitely they have exploited this particular technique for the better way. So in India, two varieties which are named Pusa Jai Kisan in mustard brassica and in citronella have been released for cultivation. So improved variety of rice also have been released through pollen haploid somaclone method, which combined microspore culture with somatic tissue culture. Then somaclonally derived mutants in tomato with altered color, taste, texture, and shelf life are marketed by USA by Fresh World Farms. So they are exploiting this technique to use in a beneficial way in a vegetables also, in a cereals also. Then production of a disease resistant variety. So this is again, we have studied, this is the third application that we are studying of plant tissue culture, right? So first also the pathogen free plant, second also the pathogen free plant, and today this also we can produce disease resistant varieties, right? So the greatest contribution of a somaclonal variation toward plant improvement is the development of a disease-resistant genotypes in various crop species. So resistance was first reported in a sugarcane for eye spot disease, which was caused by Helminthosporium, then downy mildew, which is caused by sclerospora, and Fiji virus disease caused by regenerating plants from the callus of susceptible clones and screening the soma clones, right? We need to screen the various soma clones of which one is the resistant one. So this is how they have used this for. So these are the crops where the disease resistant varieties have been generated in barley, and then in maize against Helminthosporium, in rice again against Helminthosporium oryzae, then in a rapeseed against Poma and Alternaria, then sugarcane against virus also and Helminthosporium, right? So they have been used here. Then production of abiotic stress resistant variety, right? So that was the biotic stress resistant, because it is, what is abiotic stress? Stress in a plant which is caused due to unfavorable environmental condition, and biotic stress in a plant which is caused due to the pathogen, right? So abiotic stress and a biotic stress, there are two different terms, right? So Soma clonal variation has resulting in several interested biochemical mutants which are being successfully used in plant metabolic pathway studies, amino acids and secondary metabolic pathways. So these research have shown that levels of free amino acids, especially proline, increasing during the cold hardening. And hardening in a cold is being done. So in vitro selection has been used to obtain plants with increased acid soil, salt, aluminium, and other herbicide resistance. So we can use we can develop abiotic stress resistant variety by using this technique. Then production of a cold tolerance, right? So plants which can tolerate the cold, right? So there are a group of workers, they developed Soma clonal variants for freezing tolerance in winter wheat, right? So wheat which is grown in a winter, if it can tolerate that freezing cold, and it can be grown in that particular season, right? So a significant positive correlation between proline level and frost tolerance has been observed in a broad spectrum of a genotype. So that means अगर proline level ज़्यादा है, positive correlation means if there is a more proline level, the more frost tolerance is being observed in the plant or that particular wheat, right? So in vitro selection and regeneration of a hydroxyproline resistant lines for winter wheat with increased frost tolerance and increased proline content has been reported. So the results showed a strong correlation of increased frost tolerance with the increased proline content. So as the proline content is being increased, its frost tolerance also is being increased, right? So they can do that through this technique. Then production of a salt tolerance. So plant tissue culture techniques, again, they can generate salt tolerant cell lines in a several plants such as rice, tobacco, or Solanum nigrum, or a sorghum, or a maize, right? So in most of the cases, development of a cellular salt tolerance was always barrier for a successful plant regeneration, right? Or if plants were obtained, they did not inherit. So अगर एक generation में वो salt tolerant है, तो they never inherited that salt tolerance for the next generation, right? So only in few cases it was possible to regenerate salt tolerant plants, right? So Mandal et al. In 1999, they developed salt tolerant Soma clone from indigenous rice cultivar. Now this Soma clone again yielded 36.3 quintal per hectare under salt stress as compared to 44 quintal per hectare under normal soil. So again 36.3 was not that bad if they are getting under the high salt, under the salt stress, right? So some of the reports for successful production of healthy, fertile, and genetically stable salt tolerant regenerates from the various explants have been reported. Then production of aluminium tolerance. So again, stress, basically aluminium stress. So certain soils, they could be the more amount of the aluminium in the soil, right? So if the plants cannot be grown if they are not resistant to that kind of a stress. So a lot of research has been focused on the to know what is the physiological or a genetic and molecular processes that lead to the aluminium tolerance. So despite the problems which were encountered in adapting culture media for in vitro selection for aluminium resistant, cell lines have been isolated for some species such as carrot, tomato, sorghum, and tobacco. So by this method, they have done this also. Then production of a drought tolerance. So drought is a major factor in many parts of the India. Also, there's a water scarcity, right? They are still dependent upon the rainwater, and rain is a very less in the few parts of the India, right? So they developed a sorghum Sumatran variant line which was resistant to drought stress. A novel hybrid was developed by crossing this one particular line, resistant line, with several sterile lines, which suggested that selection of somaclonal variants is an effective method for creating new sources of a genetic variation. Then drought-tolerant rice lines were also obtained in vitro selection of a seed-induced callus on media, which has a peg as selective agent, and which stimulated the effect of drought in a tissue culture condition. Like, so artificially it creates that condition. Then production of a herbicide resistance. So herbicide resistance, again, why herbicide resistance is important, because there are a lot of chemicals or herbicides we use to kill weeds or plants, right? So weed plants, they are grown around the cultivated plants also. They are not separate. So whenever we are spraying those herbicides to kill weeds or any other plants, they are definitely affecting the plants also, right? So we need to come up with certain herbicide resistance also, that they do not affect the other plants. So plants such as tobacco, soybean, wheat, maize, which are resistant to various herbicides such as glyphosate or imidazolines, etc., they have been developed. So herbicide resistance has also been developed by using this technique. Then production of insect resistance. This is important. Many insects, they affect the plant. They reduce the yield. They eat up the plant, right? Unke larvae hote hain, and then they eat whole plant, and plant is destroyed or a yield is destroyed. So again, in this, the Metro et al. In 1993, they used in vitro selection technique for generation of Somaclonal variants for Russian wheat aphid, right? This was a Russian wheat aphid in the wheat which was affected, and from a susceptible wheat cultivar, they were exposed to extract from the aphid. Resistance to aphid was observed in both R2 and R3 generation. So they used this mechanism to develop insect resistance in the plant. Then seed quality improvement. So the Lathyrus is a pea plant basically, right? The Lathyrus. So it has been identified for cultivation in Central Asia, and which has been developed through Somaclonal variation. It has a low ODAP, a neurotoxin, indicating the potential of Somaclonal variation for development of variety with improved seed quality. So they can also improve the seed quality or certain composition of a certain compounds. They can reduce their amount in a plants which are being grown by this method. So they can enhance the seed quality also by this method. Then introgression of alien genes, right? So introgression of alien genes means that there are genome rearrangements during the tissue culture. So they also provide a new opportunity for a alien or a foreign gene introgations, which can help to widen the crop germplasm. This is something transgenic kind of a thing, right? So introgations means some new genes can be introduced, right? So which can help to widen the crop germplasm base by culturing immature embryos of a wide crosses where crop and alien chromosome can replicate through a normal process of a meiosis. So introgression of a genes may be achieved by imposing one or more cell culture cycles on an interspecific hybrid material, and then the enhanced somatic genome exchange is likely to produce regenerates where part of the alien genome has been somatically recombined into chromosome of the crop species. So this can be useful in certain areas, right, where the gene that we are entering or we are trying to put it into the plant can be maybe for a better yield or maybe for some other quality which is not present normally in the other plant. So it can be done, right? Then limitations, as we have so many positive effect, though it started with a negative point that we don't want Somaclonal variation, but still we come up with so many benefit points, so many uses out of it. So what are the negative points? So uncontrollable and unpredictable nature of variation. So we still don't know what kind of a variation is being introduced, if the variation which is being introduced is useful or not. So this we need to check. So if it is not useful, that means it is not of our use, right? And most of the variations are usually not of apparent use. So we have to wait for a lot of time, and we have to check a lot of Somaclonal variant or these plants to see the variations and to check whether they are useful or not. The variation is again cultivar dependent. It depends upon the cultivar, and it is usually not always very stable and heritable. What does this mean? Maybe it is in one generation, then maybe it is not in the other generation. So that is also one problem. If it is useful variation, we would like to have this variation in all the generation. But if it is just in R2 or R3 or R2, not in R3 and R4, then it is of no use to us, right? So changes, then they occur at a variable frequencies, and not all changes are novel. in majority of the cases, improved variants have not been selected for variant purposes. So this is somewhere like a hit and trial method, that sometimes we can get variations which are useful and we can use them, and many times the probability is less, right? Probability of getting good variation is less, and getting variations which we don't need is more. So definitely one has to be very patient and keep on checking so many, all the changes in the chroma, these Soma clonal variants or these plants that we have derived are useful or not useful. So we have both benefits also, and we have both drawbacks also of this particular thing, right? So that's all from my side, and if there's anything you need to ask, you can ask your questions here. Okay, we have, okay, Vivek is able to join today. Fine. So again, I will tell you same thing. Just go through the things, whatever you're reading. These things are new for you. I'm sure you have not heard of these things earlier. Yes, there are genetic terms like DNA methylations or deletions or mutations or a polypeptide. These terms you have used, but then plant tissue culture is definitely a new for you. Right? So just go through this. Just try to read more literature about this. Read more of. You'll find a lot of papers on the internet about this particular topic. So the more you'll read, the more you'll understand, and more you'll get to know about the particular topic that you're teaching, right? So okay, so I'll stop here, and we'll meet tomorrow. We have a practical, right? Plant pathology. Okay, so I'll see you. And please make sure your practical files are submitted. Practical 1, I guess everyone has submitted. 2 and 3, I'm not sure. Last time when I saw, many were not submitted. So make sure all your practical files are submitted tomorrow before your practical class, right? So we'll see what, since the most of the practical in plant pathology are basically about the disease plant section cutting and seeing it under the microscope. So I don't know, this is very difficult, that how we'll do it here. So I'll just try to find the slides of various pathogens, how we see them under the microscope, what kind of structures we see, and then we'll try to go through that, because this we need to do it ourselves. But we cannot do it online. We need to do this offline, and these are the only 2 experiments we were having. One was the coarse postulate, and second was about the studying different pathogens under the section cutting, basically. So it is difficult right now. So if let's hope things get better in February and we get to do at least some of the practical in the lab. So we'll see how we can do it online, right? Okay, so I'll close here, and I'll see you tomorrow. Please, for attendance, you all switch on your cameras. Adhish Mishra.
Present, ma'am.
Ayushi.
Present, ma'am.
Sam.
Present, ma'am.
सैम के बाद कौन है? दिवांशी.
Present, ma'am.
इशिता.
Present, ma'am.
Janvi.
Present, ma'am.
Kshitij. Yes, good to see you, Janvi.
Present.
Manaswani.
Present, ma'am.
नम्रता.
Present, ma'am.
नम्रता को बहुत ठंड लग रही है. वैसे ठंड है. It is really very cold, I tell you. No, it's okay, Namrata. I know it is very cold. It's really very cold these days. Yes, Nehal.
Present, ma'am.
Rupal.
Present, ma'am.
Sahiba.
Present, ma'am.
Sanjana.
Present, ma'am.
Shakti.
Present, ma'am.
Shivani.
Present, ma'am.
Tanvi.
Present, ma'am.
Tridiv. Okay. Vivek. Yukta. ये विवेक को भी ठंड लग रही है. I can see that. युवराज. हाँ, युक्ता को भी लग रही है. सबको लग रही है. Don't worry. We all, except Manaswani, I guess we all are in the cold zone without any sun these days. I mean, we hardly see any sun, right? And I really hope you all are fine, right? Vivek, are you fine now?
Yes, ma'am. I'm doing much
Okay. And others, please take care. Though we always say that this variant is not as deadly as it was the earlier one, Delta variant, but still we need to be very careful, right? So all of you, please take care and don't go out unless it is required, and follow all COVID guidelines. So it's better that we enjoy winters in our homes. And I really hope that you all have a very less classes also this time, right? So you can get to have some free time also where you can think about your project and your higher education. So keep doing that. I'm just reminding you again and again this thing, right? So okay, so I'll see you tomorrow with your practical files. Thank you so much.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you,
Thank you,
ma'am.
ma'am. Thank you, ma'am.